All right. Hey, everybody. Today we're doing lab 9 and 10. So let's get right to it. Okay. Main thing that we're going to look at for blood pressure are these two values, systole, diastole. Systole being the first number, diastole being the second, typically lower number, right? Both of these are happening in the ventricle, okay? Or at least this is a measurement of what's happening in the ventricle, right? It's doing most of the work, 80%. That's what we're going to feel, right? What happens to measure these is that typically, this is how your blood's flowing right here. Laminar flow, like we can't hear our blood pounding in our arms, right? Anything like that. The way that we measure blood pressure is we use that cuff to basically cut everything off. We cut that pressure off. So that's when you turn up the pressure, let's say to like, typically a healthcare professional is going to turn that up to like 210, and then on the cuff for the pressure, and then it starts going down. Because at 210, pretty much guaranteed that nobody's blood pressure is going to be that high to break that amount of pressure. Now, as it goes down from 210, so it's like we started 210 and now we're going down to like 200, 180, 150, 130. Then boom, let's say at like right around 130 for systole, the blood pressure is high enough to overcome the pressure that was applied by the cuff on your arm. That's the first number. At this point, we keep counting. So that's our first number. As a healthcare professional is going to keep listening, that noisy, like turbulent flow that keeps coming out, you're going to keep listening until the cuff pressure is low enough that you don't hear anything anymore. And let's say that window of time is going to come down from 130, let's say to like 60 or something. And that's your measurement. Systole over diastole looks pretty good, right? It's actually a little high. So those are the values that you're looking for. You turn the pressure on the cuff up, you wait so you can hear no sound, then you wait for sound. Once sound occurs, that's your first number. You keep listening until sound cuts off. That's your second number. Then the biology obviously behind this is that it's a measurement of how much pressure your blood has built and can overcome the cuff, basically. Okay, so looking up here, there's a couple ways that we can calculate mean arterial pressure from these two values. These two values aren't, very friendly. They can't be averaged. They can't be looked at as like a single value. It's a little tough, right? First thing you can do is look at pulse pressure. This is super easy. Systole minus diastole. That's it. Super good, okay? Just like over here, not too bad. Okay, pulse pressure is used to calculate mean arterial pressure. So all MAP is, it's just a nice metric for what's your blood pressure at. That's it. And right here, we keep our normal diastole, keep that value there, and you can plug in 1/3 of the pulse pressure that you just calculated. So whatever diastole plus 1/3 of pulse pressure was, that's your MAP. If you look right here, and I think this is going to correspond nicely to that example right there, you can work out the whole thing. You will be expected to do that on the exam. Okay, so looks pretty good. This is actually a fun lab because you actually get to take some physiology data. And yeah, the machines do this quite a bit, but there will be situations where it's going to pay to know how this works and why you may be seeing strange results if you do see them. Okay, so mean arterial pressure, we know how to make this value show up. The BioPack is going to do a little bit for us as well right here. How do we know, for example, how it's regulated? We're going to test that in a few ways. So the good thing, and it does seem like a lot, trust me, there's a lot of stuff flying around with the heart right here. The good thing that I'll tell you is all these acronyms, cardiac output, venous return, stroke volume, I think TPR. The cool thing is that they're all directly related. What does that mean? If one of these things goes up, the others are all going up. If you increase TPR, so you increase the pressure in your arteries, heart rate, like all that stuff, cardiac output, that's all expected up too. So all those final values are typically always going to follow each other up, 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 up. So even contractility, where like the heart is pounding harder, right? Like we're going to see with the turtle, or like we did see actually past tense. Anything that is increasing in these values will across the board typically result in increased values in the other measurements, basically. So long as you have like a good system for recognizing biologically what's going on with each of these little things and see like, okay, like when this goes up, so if heart rate goes up, obviously cardiac output's going up. Stroke volume goes up, that means cardiac volume's going up too, or output's going up too. So the way this is measured right here, or like this is a pretty good figure right here, because it basically shows, okay, if you have high, any of these, if any of these are increasing, this is going to increase, and thus the pressure across the body is going to increase. So if the heart's doing more, that means more blood's flowing through the body. That means the pressure is going to be higher no matter what. Now there's one curveball, and it's the thing that we're actually measuring. The only value in measurement that is inverse to these is the little value that we're measuring, peripheral pulse pressure. So if you have an artery, it's got blood, hooray, we're over here. Arteries have these small, basically inlets called capillaries, and these feed the tissues blood. Now your skin, let's say, is on the border right here is your skin. We can only feel the pressure with our sensors from BioPAC on the skin. We can't plug you into the artery. Painful, gross, not good. So what we use is actually PPP, because PPP is actually measuring right here. And technically it's inverse of MAP. So when you see a high peripheral pulse pressure, it means that the blood is actually outside the artery. That means that if the blood's outside the artery, because it's being given to the tissue, that means you actually have a lower MAP. This is the only curveball for this lab. And it's typically going to be expected that you can recognize that on the exam. And it's not so much a curveball. It is a better way to test this. It's a less invasive way to test this on a patient that's sitting down. So it's not the worst thing to get used to. This is the only one where up is going to equal down. All the rest of these, if one of these is up, like I said, besides PPP, everything, the rest of them are up too. Okay. So looking at that, there are different ways to basically change all this. One of the easiest ways is going to be your body telling your arteries to tighten up and constrict and increase the pressure of blood to get blood faster to other places, or to dilate, slow down a little bit. So when you dilate things, you have a lower TPR. This is going to lower your cardiac output, and it's going to lower your MAP. Easy enough, right? Now, if you increase the pressure, you're going to have a higher cardiac output. You're going to have a higher mean arterial pressure right here too. Typically, you see a lot of high blood pressure either chronically, because a person's heart is doing a lot of work to get blood to the rest of their body. That's typically a sign of you're not doing so. You probably could use some cardio health type stuff. It's not the case for everybody, obviously. Every patient's different. But in this case, you'll typically see that relationship with hypertension. You will also see increased TPR. I believe if you're overdosing on alcohol, and you shouldn't use the word overdose, I guess, but at the same time, those of you that will encounter an emergency room rotation, you'll see a lot of your customers are going to be people that are coming in not doing super well because of imbibing too much. A lot of the cases, and that goes up and down depending on how much that's been. So maybe this example is a little too back and forth, but anything that raises your blood pressure is going to raise a lot of other stuff around your body. So it's important to understand why that happens. Typically, let's say we're not putting anything in, let's say it's something like endogenous. The way that we actually detect what our blood pressure is that we have baroreceptors, little stretch receptors that say like, okay, like how stretched out are we? Should we constrict? Should we dilate? Their job is to regulate your body without you telling it to, right? Because again, this is one of those automatic type systems, involuntary. So there's a few ways that the nervous system can actually control this. And this is a nice list, and I always highlight it because they're kind of ranked in power. Like, how much commitment do you want to make to increasing your blood pressure? So if we need to get, a higher MAP, and the example up here is talking about if you have a massive blood loss, like a hemorrhage, so like somebody from a car accident, you need to get a lot more blood flowing because you've lost a lot of that volume, and like it shows right here. If you lose a lot of volume, can't be pumping as much blood. That's SV. You're gonna have a lower output, you're gonna have lower pressure. Your body's gonna try and raise that a few ways. Lowest stake way to raise blood pressure, increase the heart rate. Just get some adrenal receptors going, right? We're just gonna get those pumped up, norepinephrine, let's go, go, go. Maybe you don't even go that far. Typically your body's just gonna be like, all right, increase the heart rate, we need more cardiac output to overcome this, right? Next, It can constrict your arteries. That's going to increase the pressure, right? Smaller arteries, higher pressure in the blood. Blood can flow a little farther, even when, in the case of the hemorrhage, a lot of that blood's gone, right? Venomotor tone is going to be a little bit different. This is a higher stakes way that your body has to commit to basically getting blood around. Basically, venomotor tone is going to be your body's smooth muscle is going to kind of contract and leave some, at least increase venous return in this case. So that you can get more blood kind of pushing back and forth in that case. The highest commitment that you can do, and remember, these are all from the sympathetic nervous system, which is like go, go, go. Contractility, you can tell the heart, fire harder, pound harder, basically, and get me more blood everywhere. That's tough. I mean, I think contractility from, what I read is always like, it's not like a final stand or anything. Your heart can turn to that quite a bit. It's just more like you're asking more of the heart in that case, I'd say. All right, so parasympathetic, the way that we would increase MAP is we would actually tone down the parasympathetic effect. So we would not want to see acetylcholine in times of a need for higher blood pressure. We would not want that. So parasympathetic is toned down right here. Thus, if you have a toned down parasympathetic, you'll have a higher heart rate, MAP, cardiac output, whole thing. Okay, so for Valsalva, this can be tricky. So I always typically go through the data with everyone. So first things first is that you're measuring your PPP right here. We're not technically measuring straight up MAP. So right here, PPP, it's the opposite of MAP. So if this is considered high, then technically what we're looking at is a lower MAP. Okay, so let's practice this on the first set of data. And remember, when you guys take the data, it can be a little different, so don't worry. There's a few stages in this technique that we're going to do, and I'll allow you to read up on the Valsalva. Basically, you just like hold your breath and like strain to try and breathe out, but you can't, and that's going to increase a lot of pressure centers, things like that. Okay, so as we're going here, here's your PPP. It looks pretty good. Everything's fine. Okay. As you begin this technique, and this is something used, you have an initial spike in PPP, and then a massive drop, kind of a correction, and then we end the technique and we kind of come back, right? So there's three distinct things that can happen here. I don't know which one to use. Okay. There's the beginning, there's the correction, and then there's the end, right? So if we have a high PPP at the beginning of this, what's happening is that at the beginning of the Valsalva, your chest cavity has lost a lot of volume. In that case, you've taken a lot of that volume away. That means that if we're looking at, this is a trick that I use to keep track of MAP. If MAP is down here in red, and all of a sudden you have a really high, oh no, give me that back. I'm gonna do it inverse up here. So if you have a lower venous return, you're going to have a lower return and a stroke volume with your heart. What that means is you have a lower cardiac output in this little number one moment at the beginning, before the correction. If you have a lower cardiac output, you have a lower MAP. So that means if your MAP is inverse, remember, to PPP, your MAP actually drops. Your mean arterial pressure drops in that moment that you're doing the technique. And that's why it feels so bad, right? It's like, oh no. But then as PPP drops in blue, MAP overtake is a lot higher. Your blood pressure is higher now because it has to correct the response that you just gave it. Now, as you end the technique and as you recover, MAP goes back down. Now, this isn't an exact illustration. I do try and just show that there's going to be a response between these two for the technique. So when you begin the Valsalva event, yeah, your PPP is going to push all your blood into the capillaries. That means there's less blood in the arteries. That means your MAP is lower. So that's why we see this downward spike right here. But the correction that your body makes through the sympathetic nervous system and by toning down the parasympathetic is that you're going to increase that MAP back to sustainable levels to get your head back above water. Yeah, and I mean, if I was really, I shouldn't have done the inverse so drastically, like right here. It's probably just going to be like back normal like that. Okay, now you don't have to draw any of that. Typically, it's just a way for me to make sense of all this. Something similar is going to happen with the sitting and standing. So if we're sitting, everything's good, and then all of a sudden we're standing, you see there's a drop. Because right here, pulse pressure is going to go down. That means if we're looking at MAP, once we stand, it's kind of like a tree. You have to push the blood up back to the head, get back up there. But then as you're sitting again, things kind of go back down. Okay. So in response to standing, you get a higher MAP because you do have to force that pressure to get blood back to the head and feed the brain, right? We talked about it's kind of important. All right. So hopefully that's a good little intro. At the same time, you'll get a lot of time to just actually do this stuff in lab. So it should be good. All right.